From Microbe TV, this is Office Hours for Wednesday, the 15th of January, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can talk about viruses and many other things as well. Welcome to our moderators tonight. Let's see who's here. We have Les. We have Les. We have Les. We have Les. And we have Les. Seems like Les is the only moderator here at the moment. That may change. Thank you, Les, for being here. And thank you all for being here. Tell us where you're from. We could give you a shout out. What do we have planned tonight? We're going to talk a little bit about the YouTube takedown of Beyond the Noise 48. And then we'll have a mini lecture on genomes and genetics. Chris and Lisa Lebowski from snowy Montreal. Minus 11. Oh my gosh. That's really cold. Next week it's supposed to be minus 15 C here in the New York area. That's crazy. Philip is in Wales. Minus 2 degrees C at the moment. Patty is in Massachusetts, minus 6 C. Dag Finn is in Oslo, Norway, 2 degrees and foggy. It's 2 a.m. You're pretty brave to stay up at that hour, Dag Finn. Brian Silverman is in Montreal, minus 10 and mostly clear. Will is in Changsha. Of course, that's in China, where it's 1 degrees Celsius. Dean is from Wisconsin. Ray's is from Northern California. Anyone from California here? In particular, around the LA region, and hopefully you're doing okay. If you're from there, you're probably not. Excuse me. You're probably not watching Office Hours. Jonathan is in Encinitas, where it's 17 C and dry. Rima, hello, from the middle of nowhere. Okay. Dean, hello from Wisconsin. Abhilash is from Frankfurt, Germany. Maureen is in snowy and cold Northeast Ohio. Welcome. Eliza, Southern Ontario, minus 6 and a bit of snow. By the way, here, what is it here? I'll tell you. My phone is under the drive. Come on. What do we got? Currently, it is minus 3. Out in New Jersey, where I am, minus 3 Celsius. My Jessica's a mess. It needs cleaning. Julie is 27F in north central Pennsylvania. John is minus 3 C in Minneapolis. Kang is in Chicago, minus 9. Truman 17 is in Fort Worth, Texas. 3 inches of snow last week. Wow. 11 C currently. Jessica is in Toledo, Ohio. 9F tonight. Pete's in London, 8 degrees C. Cold spell has ended. Yeah, so Beyond the Noise 48 is finally back up. I'll tell you the story about that. Troy is from Oklahoma, 7 degrees Celsius. James is in Longmont, 7 C. Hypnicity is in Linwood, Washington. Dagfin, I'm not brave to stay up. I'm hungry to hear a reasonable voice from the internet's best science informant. Thank you very much. I appreciate your confidence in what we do. We deal in the truth. That's it. Not misinformation, for sure. So as you can imagine, to be told we are peddling misinformation is quite offensive. Hello, Betty from Washington Heights. Spamla is in Bedfordshire, UK. Heather is in Kamloops, BC, 1 degrees Celsius. Nebby is in Central Florida, where it's warmer than anyone so far, 60 C. Logotic is in Chicago, -9 C. Pat is in Nova Scotia, -4 and cloudy. Hello, Barb Mack is here. Welcome, thank you. Another moderator. Appreciate your being here. And yes, Microbe TV is on Blue Sky. Go check it out. What is the address? Blue Sky. microbetv.bsky.social. Okay? That is apparently it. I do not spend a lot of time there, but I plan to start spending more time there. Kim, our social media specialist, does most of the heavy lifting over there. Well, thanks for coming, everyone, tonight. First thing I want to tell you, so you remember last week, Beyond the Noise 48 goes up, we publish it. It's about what questions you would ask RFK Jr. during his confirmation hearings. So basically, pull off it. Says some of the things that RFK Jr. have said over the years, which are medical misinformation, by the way. And then I asked, well, okay, what kind of questions would you ask him at the hearing? Same day, it gets taken down for spreading medical misinformation. So I appeal. I presume it was just a bot or someone being a jerk. A couple of days, I appeal immediately, which only involved pushing a button saying appeal. You weren't allowed to write anything. A few days later, I think a Saturday, they say, we're sorry, your appeal has been turned down. The video remains offline. We hope you understand. No, I don't understand at all. And since I can't talk to anyone, it's extremely frustrating. So now I'm trying to figure out what to do. I put the video on Vimeo, by the way, which never takes anything down. But it doesn't have the same reach as YouTube, right? But at least we could get it embedded on the webpage. I'm very frustrated because we're going to do more Beyond the Noise. And, if I get another one taken down, then I can't publish anything for a week. And that's bad. So misinformation wins. Then suddenly, yesterday, boom, it's back up again. People are telling me it's up. I said, I don't know anything about it because nobody contacted me. Nobody said, oh, Mr. Racaniello, we're so sorry for our error. It's up and the strike against you has been removed. Anyway, it's up. I don't know what happened. I would really like to know what led to the takedown in the first place to try and avoid it, right? If it was a bot or if it was some listener who decided they don't like what we're saying. But, we repeat RFK Jr.'s misinformation. So people may think we're spreading misinformation by doing that. What we do in the videos, we say RFK said this. But if you don't listen to that part, you're going to think we're just saying vaccines have not been shown to be safe and effective or something like that. That's a quote from RFK Jr. Problematic these days. People not listening. I don't know what the answer is. But as Les says, AI is heavy on the artificial and light on the intelligence. The problem is it's big and it's hard to check everything. And that's probably why Zuck stopped fact-checking over at Facebook and Instagram, right? So now you don't believe anything you read there. So anyway, now Beyond the Noise 48 is back up, and Patricia listened to it and liked it. So we have also now broken it down. So there were 8 questions. Let me put the right number of figures up. I don't want to spread misinformation. There are 8 questions that Offutt proposed. And so we have broken the original video, which is 30 minutes, into 8 one-minute brief videos which state the question and why, basically. So we're going to put those up as well. The hearing will probably be at the end of January. So we want to get those up before the hearing goes so people can know what's at stake. Genster says takedowns are often the result of multiple reports by people with opposite views. Probably an RFK fan. Yeah, the problem is Genster. RFK Jr. is all over YouTube with misinformation. He doesn't get taken down. Rogan, Friedman, all kinds of other podcasts. Well, why is that? Why don't the bots pick that up? Why don't like last week, I asked everyone to go flag those, and they're still up for misinformation. They're still up. So much for that. Anyway, so the bottom line, folks, is hello, Planet Steel. Welcome from Scotland. Bottom line is Beyond the Noise 48 is up. You can go listen to it. You can go watch it. And we recorded 49 on Monday, and I'm going to release it soon, tomorrow, or is it even edited? Let's see. How can I find this? Oh, yes, we will look for, see if it's edited. See if my editor has edited it yet. Microbe TV shows, Beyond the Noise episodes, 49. Yeah, 49 is up, is edited. So, can I post it tomorrow, perhaps? Yeah, I think I could do it tomorrow. I have an all-day textbook meeting, but it should be done around 3, 4 o'clock. You need an espresso. Yeah, I don't know why I'm yawning. I'm sorry. I haven't felt tired all day. I'm fine, so I don't know what's going on. Maybe it's too warm here. Yeah, I could use an espresso, though. Raise11 says, we're all ADHD thanks to our phones. It's one reason why no one listens past the question. Yeah, it's really unfortunate that there's a minimum of listening. I'm sad about that. I see it all the time. Our RFK Jr. fans welcome here. They can come. This is free. This is open. You can come here, but you can't spew misinformation in the comments. All right, we don't want that. You're going to get kicked out if you spew misinformation. You're welcome to sit here and listen and maybe learn something. But if you follow, by the way, the latest in RFK Jr.'s book about Tony Fauci, which is huge. It's about this thick, and it's all misinformation about Fauci. He says that he doesn't believe in the germ theory of disease. He doesn't believe that bacteria and viruses and fungi and parasites cause diseases. So just go away. That's what I think. Go away, because this is nonsense. It's a waste of our time to even deal with it. And this person shouldn't even be considered for this position. And the senators have sense, and hopefully some of them do, and they don't think politics. They will vote against his confirmation. This is a joke. It is a joke if he thinks the germ theory of disease is wrong. He, on his own, and all the scientists feel otherwise. Ay, ay, ay. Dagfin reported the Rogan RFK podcast. It's still up, you see. I'm going to take Rogan down. Spamlet says, what are they going to do when RFK Jr. makes official misinformation posts? So if he's confirmed as head of HHS and he begins to spew nonsense, people are going to just have to object. We're going to call him out here for sure, and other people are going to have to object. Absolutely. Yeah, that could happen. But the Surgeon General of Florida spews misinformation, right? He says mRNA vaccines integrate into your genome and cause disease. Well, they don't integrate. They haven't been shown to integrate into your genome. In cells in culture, under certain conditions, they can be reverse transcribed to DNA and then integrate into your genome. But that's not a human. Okay, and now these vaccines are in millions and millions of people, and we haven't seen any problems. So stop the misinformation, Surgeon General of Florida. The unfortunate situation is then the people of Florida don't know what to believe. They're not scientists. They have this Surgeon General who's eminently educated, right, saying these things. What are they supposed to think? He's wrong, he's right, they don't know. So that's where misinformation podcasts take advantage. They can immediately get out something and say, see, we told you so. The Surgeon General now says it's so. The vaccines are not good. That's his statement, by the way, not mine. So it's already a problem. Yeah. Patricia likes to count the number of duck decoys. I don't know anything about that. He goes hunting. Emily, thank you for your content. You're welcome. My pleasure. Try to keep things correct and civilized. John writes, Vax Talk, Voices for Vaccines podcast, had Dr. Offit on their latest episode 2. I always make time to listen to any interview he's on since finding him through Vincent. He is about the truth, information, right? He just tells you what it is. He doesn't obfuscate the truth. And that's why I like him. Jeff writes, the reason that certain misinformation is allowed is purely political. The GOP and their supporters have been successful in making believe that they are targets of censorship. I don't know why certain podcasts are not taken down when there's a lot of misinformation on them. It could be political, which is sad because YouTube shouldn't stoop to that. And then here, in this case, Microbe TV, we're totally correct information. Except maybe we make a mistake now and then, but we correct it. We don't purposefully spread medical misinformation, but RFK Jr. and many others do. I don't know what kind of world we're living in right now. It's ridiculous. Is this specific to the U.S. or does this happen other places? So people from other countries, what's going on? Is there misinformation in your country too? YouTube is an international thing. I guess there is, right? Yeah, they have always been successful in making people believe there are two sides to every point. Yeah, right. Everybody's position has to be heard. That's the Friedman idea. Both sides. When they're not two sides, right? Vaccines work. They save lives. That's it. There's not another side that needs to be heard because the data do not say that. By the way, did you see the FDA just voted to put a warning in the package information for the RSV vaccines that they may be associated with rare events of Guillain-Barré syndrome. One in millions. They're not even sure, but just to be cautious, so you can make your own decision. If they didn't care about vaccine safety, they wouldn't do that. So just don't give me this stuff. Now, if something is 1 in 9 million. You're never going to pick it up on a placebo-controlled trial. You have to license the vaccine and then do post-marketing studies to see. And that's what's been going on. They've been monitoring the studies, the safety of that vaccine ever since it was licensed. They pick up a little signature of Guillain-Barré. What's Guillain-Barré? It's an inflammation of the nerve. It usually starts in the lower extremities and moves up. It can be painful. Sometimes you get paralysis. Usually recovery is complete, but not always. Hello, Tom. Welcome, our moderator. Good to see you. And Jill from Wilmington, Delaware. 26F. Clear tonight. Wayne is in Urbana, Illinois, where it is 13F and 11C. Dagfin writes, what is going to happen when the authorities supposed to oversee that treatment is safe and effective, start changing treatment guidelines, replacing safe and effective treatment with ivermectin, etc. Yeah, well, that's one of the things RFK wants to do. He says ivermectin and hydroxychloroquine work. We should be using those. What? They don't. There's no evidence that they work. I don't care who says they work, what anecdotal story you have. Oh, I took ivermectin and my long COVID went away. Great. I'm happy for you. But it doesn't mean that it's ivermectin that did that. You have to do a proper study. But yes, I hope that, look, the chance that he gets confirmed, I hope sane heads will prevail at NIH and other institutions who will say, no, you can't say that. You can't take out the vaccine and put in ivermectin. Give me a break. We have a big science community. We have one RFK Jr. I hope the science community can prevail. Ay, ay, ay. Minus 11 in Urbana. That's better. Very good. Hit the like button, folks. We have 140 people here and 55 likes. Please hit the likes. Why? Maybe we attract some people who want to learn something, right? Not disrupt, want to learn. Disruption is not the way to go. Okay, so Pete writes, what's the purpose of nominating RFK? Purely to create chaos, confusion, kicking the dust to disrupt policymaking. Okay, so there are two possibilities. First is that Trump actually thinks RFK is into something, right? But the fact that RFK wanted to get rid of the polio vaccine and Trump said, no, you're not going to do that, that should be a little alarm for Trump, right? And the other is that, he wants to do a Saturday Night Live skit and just be silly. He wants to break everything, declare martial law, and take over. Well, that's what I've heard. I don't actually believe that. But he thinks these people are all good. I mean, all of the health and science nominees are flawed. I've gone over that in the previous office hours. And he just thinks they have a different view. It's contrary to what everyone else has, and therefore it's good. And that's wrong, right? It's really unfortunate. After Rogan and Mel G spewed about ivermectin curing cancer, it's clear Rogan has a free pass. Yeah. I mean, I just don't understand. YouTube, you have an algorithm. Why doesn't it pull down Rogan and Friedman? Huh? What kind of an algorithm is that? Do you know how to use your computers? There are two sides to every point. Being right and wrong. Yeah. We don't have to hear about the wrong side. We can say, okay, the ivermectin and the hydroxychloroquine don't work. There's no data to support it. End of story. We don't have to have someone on a podcast to support that they work, because they don't. There's no data that supports that they work, right? So I agree, but I don't want both sides on an interview. You don't need that. You don't need, if you're going to have an interview about vaccines, you don't need someone that says they've never been properly tested and they don't work. No. That is wrong from the get-go. Sorry. You can have some other thing that's debatable. That's fine. Then you can have two people. But not something as clear-cut as that. Nope. No. If I had YouTube, that's the way I'd run it. Patricia writes, a number of times I've had to screen for Guillain-Barré. Oi, no one has ever answered yes to the questions. So Guillain-Barré, there's a low continuous rate of Guillain-Barré throughout the U.S., and occasionally it's caused by vaccines or infections. So the 1976 swine flu vaccine did cause Guillain-Barré. And that's the last vaccine now that I can recall doing that. But there's always a low level, and so if you have 1 in 9 million Guillain-Barrés with RSV vaccines, it's hard to know if that's the vaccine or if that's the low level. Arthur said she hit the like button. Going back to the presidential address, we'll watch later. Political engagement is required to stop RFK. Okay, you can go watch the address. It's fine. Are we going to break down the congressional report on COVID? There's a lot of misinformation in it. Well, in our year-end summary of 2024, I went over that report to the extent that's worthy. You know, the report is much more than just COVID origins, SARS-CoV-2 origins. It has to do with masking and vaccine distribution and so forth. But there is a section on SARS-CoV-2 which simply repeats the same old tropes. They were doing gain-of-function research in Wuhan. That's the best evidence that the virus came from Wuhan. Seriously? That's ridiculous. First of all, you know, gain-of-function is not what these Congress people think that is. They think it's all bad. In fact, most research, biological research, involves gain-of-function. And yeah, they were doing gain-of-function. So what? There's no way that SARS-CoV-2 came from that lab. We have no evidence, and we have plenty of evidence otherwise. Anyway, Troy, we did go over, I went over the 5 points, which are the main points. That's the last episode of TWIV that was released. More than 2024 viruses. Yeah, both sidism has been rife in climate science reporting too. It's ridiculous. Fabricated no climate change, right? It's quite clear. It is quite clear that there is climate change. And that the people fabricating that it's not making up data, they have another agenda. And we have to hear both sides. And all it amounts to is you're lying who says climate change is real, and I have data that says it's not, but your data is actually bogus. It was collected in a way to show that there's no climate change, which is wrong. Lollipop. So, yeah, the Offit video 48 Beyond the Noise is back up. I had appealed the takedown. They said, nope, we don't believe your appeal. And then suddenly it went up on Monday with nothing done from me. So it's up, and I don't know why. So it is good. Jeff writes, regarding Guillain-Barré, if the vaccine may cause it, wouldn't the infective agent also? If it is an autoimmune disease, then it seems like an agent would cause it. Yeah, well, for the 1976 swine flu, we can't answer that question because there were no cases of swine influenza that year. It was just swine flu vaccine. So does RSV cause Guillain-Barré? Certainly a number of people who get RSV disease get Guillain-Barré because, as I said, there's a low level of Guillain-Barré in the community. But it may also be the way the vaccine is prepared. In some way, the immunogen reacts in some way to cause Guillain-Barré. So it need not necessarily be also the infectious agent. I'm thinking, RSV vaccines are spike proteins, and so they're adjuvanted, and so could have a different interaction than the virus. Hello, Carol, our favorite nephrologist from California, where it's 59F. Welcome. Bakersfield is far away from LA, right? So I guess you're okay. Bakersfield is more interior, right? Yeah, it's really hot there. Patricia writes, I guess a lot of people are going to have to ask if they can recommend stuff like ivermectin or get fired potentially. Well, hospitals have to have policies, and the physicians have to follow the policies. If a physician is not associated with a hospital, well, at some point they may have to send patients to a hospital, and they could lose that privilege if they prescribe drugs that are not approved. I don't know. This is a very tricky question, right? Don't recommend ivermectin for viruses. Recommended for parasites, for which it works very well. Doesn't do anything for virus infections. Abilash says, so I asked, is this nonsense going on in other countries? Of course, the AfD have turned East German against vaccines and are getting endorsements from Musk, who doesn't know anything about science, by the way. Maybe materials science and batteries, but not biological science. Dagfin writes, the most common cause of GBS is infectious disease. Let's see. Yeah, Campylobacter is a big cause, but also surgeries can cause it as well. But 1 in every 1,000 people with Campylobacter, it's a bacterial infection, develop GBS. Yeah, a number of viruses can trigger GBS also. In fact, vaccines inducing GBS are pretty rare. But yes, I would agree with that based on what I saw, Dagfin. John writes, Senator Klobuchar was clearly against RFK Jr. in a recent interview, and I imagine Senator Tina Smith is too, but I still wrote in that letter so they know constituents support their stance against him. Yeah, I put the link in some time ago to write your senator. And you should do that. Even if you're in a state with 2 Democratic senators, you just don't know how they're going to vote. FSM is the dog writes, John Oliver did a great video pointing out how bad both sidism gets you by having 97 scientists debate 3 deniers over climate change. Yes, it's not useful. In some cases, both sidism is useful, but not in all cases. Interesting question from Kang. If RFK Jr. was a doctor, would his license to practice be revoked? Well, it depends what he did, right? If he were an internal medicine doc and gave vaccines, and he refused to vaccinate, he was a pediatrician, even better. He refused to vaccinate his patients. Yeah, his license should be revoked. But I don't know if he would get into medical school and pass the boards and so forth, because the things that he says reflect a problem in understanding reality, facts, right? But I suppose if he were a doctor, he wouldn't be acting this way. Although, there are doctors out there who are decidedly vaccine skeptics, right? I don't understand why. I don't get it. I mean, tell me why you're a vaccine skeptic. And don't lie. Don't use misinformation, okay? Hello, Joe from Southern Ontario. Welcome. Zainul is here for the first time. His or her PhD thesis on quasispecies. Was wondering what's a burning question you've had about them in viral infection and transmission. I love quasispecies. I love them, and I didn't understand them when they were first discovered in 1976. So what is a burning question about them? So when you are infected with a virus, you usually get a very small dose. So it's a bottleneck. And so how long does it take for the quasi-species to regenerate? And would it be the same quasi-species as when you started, or are there going to be differences because of selection? I think that's an interesting question. Luis, 20C in Mexico City. Welcome. Margot, 65F in Phoenix Metro. Welcome too. John writes, Rogan and Gibson didn't understand monoclonal antibodies and how resistance can nullify them. Rogan said they pulled them to sell vaccines, among other extremely ignorant statements. Nobody pulls anything to sell anything. It's just idiocy. The monoclonals didn't work anymore. Yeah, because the variants were resistant to them. So if you don't know that, you shouldn't even be talking about antibodies and vaccines. This is a problem when a guy like Rogan talks about science. He doesn't know what he's talking about. On both sides, Will says the current journalism has lost its number one purpose, the search for truth. Well, there's certainly many journalists out there who disdain the truth and don't even talk about the truth. And that's mostly in politics, right? But the certain news stations even spew misinformation about vaccines. Yeah, so this is really bad. Tom says, 5 years into the pandemic, trying to recall a live stream remark. Amy made that, that's right. That it would be 5 years before COVID set to a pattern. So she said the pandemic would be over in 5 years and that it would establish a pattern of winter peaks, which is what's happened so far. But there are peaks elsewhere in the year, which is weird. Like this year, we had a peak earlier. Now it's going up again. So I don't know what that's about. But there's certainly a peak in the winter season. You know, your typical respiratory virus infection season. This is a good one. Abi Lash, is RFK Jr. against animal vaccines? Poultry, swine, and cattle. What are his thoughts on that? I haven't heard him talk about it. I don't think he cares about animals. He just hunts them, right? He's interested in people because he can get the most attention that way. I'm not aware that he has made any comments on animal vaccines, but please, folks, correct me if I'm wrong. Tagfin writes, does he even know they're being vaccinated, right? But you know, RFK Jr. got all his vaccines. Paul Offit said he was having a party one year and he made everyone get vaccinated. So here you say COVID vaccines are the most dangerous vaccine ever. That's a quote from RFK Jr. And then you make your guests and yourself get vaccinated to have a party. It doesn't take much to find the cynicism in this person instead of just believing him. And yeah, here's the rub. Rogan has 10 million viewers, so the video is not coming down. And that is pathetic, right? It's pathetic because it means your fact-checking is not really fact-checking. It's only for certain people. Tom writes, hard to believe the Senate Dems didn't call good virologists to testify. What are you talking about? The congressional report on COVID? Yeah, I'm very surprised. There was one minority witness who was an infectious disease physician. Like as Brienne said on Twitter, unless he is Daniel Griffin, doesn't likely know much about basic virology, which is what you need to know to talk about origins. Hello, Hans from Ecuador. There have been various studies showing SARS-CoV-2 replicates in astrocytes and neurons. No, not in someone's brain. There's inflammation, but you can make astrocytes in culture, you can make neurons in culture and get some replication, but no one has found it replicating in the brain. I haven't seen good data to support that. And Hans, that's what I look for, the data. And if you see a couple of, you do sections of the brain and you hybridize with viral probes. If you see a couple of places lighting up, that's not evidence of replication. Sorry. Um, so I don't think there's evidence that it gets in. I think there's hypoxia and there's inflammation, and that accounts for the CNS symptoms. Well, Raise 11 says people shouting stop gain of function while petting their cats and dogs. Is there some gain of function? I suppose you're breeding for certain traits. Yeah, that would be a gain of function. Yeah. Patricia says, I'm talking about the FDA, Vincent. I don't remember what we're talking about now. Let me go back and try and find it. Patricia. I guess a lot of people are going to have to ask if they can recommend stuff like ivermectin or get fired. No, at the FDA, you have a committee that votes to license something or not. It's not up to one person. If one person out of 20 wants to license ivermectin, it doesn't get licensed. Now, if they're speaking on their own and saying I'm from the FDA, then they're going to get fired because they can't do that. Yeah. All right. Yeah. Saban got Guillain-Barré. Both recovered. Yeah. Saban told me that it was very, very painful and he was paralyzed for a while, but he fully recovered. Yeah. Just found 2 literature reference case reports linking RSV infection to Guillain-Barré syndrome. You have to be careful. It's really rare. What are you going to do? I got RSV and I got Guillain-Barré. Maybe you would have gotten it without RSV. I don't know how you can do anything but making an association. So say linking may be too strong there, Tom. Rob, thank you for your support of science communication. I appreciate your donation. Parasites hate ivermectin because it works. Of course. Of course. John writes, didn't Peter McCullough get revoked recently? The cardiologist pushing things like ivermectin. I don't know, but he should be. He shouldn't be pushing ivermectin. There's no evidence that it works. There have been many clinical trials, Dr. McCullough. And just tell me they were all done incorrectly. Sure. Yep. Yeah, this skeptic, cynic. So pull off its Times column, right? Today, I guess. He said RFK Jr. is not a skeptic. He's a cynic. I don't know if he's just a liar. He says they don't work. It's not true. It's just not true. He said polio vaccine has killed more people than not. It's just not true. Hep B vaccine. He has said Hep B vaccine doesn't work. It's simply not true. So it's not even a cynic. It's just a liar. You're saying things that are simply not true. I don't know what to do about that. Hat says, see the comedian Bill Burr's takedown of Rogan on Rogan's shows. Told him to his face that he's crazy if he thinks a guy with no degree is telling the CDC what to do. Yeah, and that gets Rogan a lot more views, right? So what does Rogan care if he calls him crazy? That's what his audience loves it. Yeah, call Rogan crazy. We all love him. Yep. Interesting. Gee. Any benefit in getting the swine flu vaccine? And now you're talking about 1976, right? You were 21 and a group of you lined up at the local Kmart. No, there was no benefit because this was a bona fide swine virus. It wasn't a human virus and never spread in the human population. So no benefit that I can tell. Probably no harm, though. But those of you may remember, Gerald Ford was president, and he said, okay, we're going to do this because he was advised by a virologist to have this vaccine made and give it to everyone. He said, okay, we'll do this. And he got a shot on TV, you know. Margot writes, I've tried to figure out why my NP coworker doesn't like the COVID vaccine only. She's very, very smart. I think it's more that she's dealing with nursing home patients only. I just roll my eyes. Well, she's obviously not very, very smart if she doesn't like just the COVID vaccine. She has a bias which has no basis in reality. Okay? It doesn't like it. There's no place for not liking it. Look at the data, and then you can decide whether it is safe and effective or not. Okay, Raise 11 from my ex, an MD. She thinks vaccines are a body autonomy issue. You have the right to decide what to put in your body, and parents the right to say no. Tough argument there. Yeah. For all vaccines, the pediatrician says to the parent, I want to give your child this vaccine, okay? And most parents say, okay. Yeah, I'll take autonomy. Give me the vaccines. I know they work. Most people can't make the decision, though. Public health doesn't work if you let people make decisions who are not informed. And nowadays, they try and get informed by going online. They get misinformation, and this is a big problem. The pediatrician sits down with them and says, okay, I'm going to give your child an activated polio vaccine. This vaccine has few side effects and prevents poliomyelitis, which we need because poliovirus is circulating in the community. And it's in the U.S., it has drastically reduced the number of cases. Can I vaccinate your child? You have to say yes to that, right? Sure, make a decision. But don't say no because you feel you haven't been given a decision. That's just stupid. MDs take an oath, do no harm. Letting the patient decide no is doing harm. Because if they say no, the physician should say, okay, let's talk about why you don't want this vaccine. Oh, I heard it causes cancer. Be prepared to discuss why it does not cause cancer. And don't worry about your 15 minutes running out. This is a human being that you got to deal with here. John, sidebar on Tweevo, 76. It's been excellent. I've seen the year 2020 from so many angles through Microbe TV, and each time I learned something different. Donated via the link on the screen today. Thank you, John. We appreciate your support and your enthusiasm. Welcome, Patricia. It's okay to be late. Okie dokie. Patricia writes, the fellow from North Carolina who worked on the COVID vaccine and was an early TWIV guest would be a good witness at RFK confirmation hearing. Ralph Barrick. They better get some good people who can ask tough questions. Yeah. Kang writes, what would informed consent mean with RFK Jr.? Well, he says, RFK Jr. contends that parents are not given the information they need to make a decision, which is wrong. It's just not true. They're given all the information they need. You can go to the CDC website. You can read what their recommendations are. You can read the package inserts to find out what the side effects are, the efficacy. All the information is there. Yet he is saying we have to make sure parents have the information they need to make decisions. You're wrong. You're saying there's a problem where there is not a problem. All right? And people get confused, and so they think there's something wrong with the system. Yeah. I don't know what informed consent would mean with RFK Jr. If he really is serious about giving them information, then the doctor gives him a book and says, read this before you come to your vaccine appointment with your kid. Not happening. They're not going to read it. They're not going to understand it. But currently, the physician cannot give a vaccine to a kid without the parent's permission. And an adult, you make your own decision. If the physician says, I think you should get a pneumococcal vaccine, and you say no, they're going to argue. Why? This is a very serious disease, and you're in the age group where you should have protection. What is your objection? It doesn't take very much. Well, I heard that, no, I don't mean to put a Southern accent on. I heard that it does this. Are the physicians ready to counter? I don't know. That's a good question. Pete writes, like John, I've sent $50 to Microbe TV this week on the direct link to minimize costs and fees. Thank you very much, Pete, for your support of science communication. So just to explain, folks, Microbe TV is a nonprofit entity. We do not sell anything. We do not run ads. We get the money we need to make our programs from listeners. And if you donate, and many of you do, and it's wonderful, we can keep making programs. They cost money to produce. There's all kinds of costs associated with it. And if no one contributed, then I would have to leave the incubator and get rid of all of our contractors and just do it all myself here at home. And I don't know if I could do that. I could probably do it for TWIV, but that's about it. Verinika Privyet. Moscow, it's 5 a.m. minus 4C. Yes, so you can watch tomorrow. Thank you for coming by. Jeff writes, a lot of people are skeptical of things they have no understanding of. They don't know what they don't know because they never studied this subject. Yes. And then the skepticism is reinforced when they go online and search for vaccines, and the top 10 hits are medical misinformation because the anti-vaccine community is so effective at putting its message up first. So, yes, it's a problem with understanding to a great extent, and the anti-vaxxers take advantage of it. Julie S. writes, I wonder if RFK Jr. has ever sat at the bedside of someone dying of COVID or flu or RSV or polio. Probably not. Yeah, Paul Offit has asked the same question, right? Someone dying, and maybe because they didn't get vaccinated or maybe the pre-vaccine era to emphasize. But he may know this and just doesn't want to admit it. Tracy from California, welcome. Nice to have you here. Sergey writes, I was in New York City in May last year. I wish I could meet Vincent, but didn't dare. Yeah, you can. You can Send us an email at the incubator at microbe.tv and set up an appointment to visit. Many people do that. We're happy to have you. Gwen, I know Gwen. I've known you for many years. Sorry, I keep forgetting. I'm going to write it down. A pile of stickies again. G. Ferraro. Gwen. I'm really sorry. I always say G because I don't want to keep asking you. But now I know it's Gwen. Maybe I will remember. Tracy's getting Tdap tomorrow and the pneumococcal a few days later. Very good. That's the right thing to do. John says, if someone says they follow Dr. Offit on Twitter and then won't take vaccines, then they didn't understand a single thing they listened to. Okay. I agree. Artemis writes, joke. A man in a train makes paper balls, and throws them behind him. Why? To protect himself from tigers. Another man says there are no tigers in the trains. The man replies, see, it works. Yeah. You could make that joke up in a lot of different ways, right? Michelle is in Philly. Saturday, I went to MRC planning meeting for the 2025 events as FIFA, July 4th, possible virus, etc., that could come into the country. I see. So yeah, the FIFA. So there's going to be some soccer thing, and you're worried about infectious diseases coming in. Yeah. When is this going to happen? July 4th? Summertime. So winter respiratory infections not likely to be a problem. But there are things that can be done to mitigate this, and event planners of that magnitude know that. Greg, hi, I've been listening since before the pandemic. Engineer by training who has done statistics for 30 years. Looking at examples of paper peer reviews is how I found TWIV. Very informative. Thank you so much for doing that, for being here and learning. Yeah. Lillian wants to know if I'll watch the Senate confirmation hearings. I'm sure they will be recorded. I can't watch them if they conflict with the podcast recording. But I would like to record them because if it gets frustrating, then I can fast forward. Gang writes, in the episode about discophage testing, is there an advantage for bacteria with plasmids that make them vulnerable to phages? Any phage for bacteria with plasmids coded for antibiotic resistance. So the advantage for the bacteria is that these plasmids can have antibiotic resistance genes on them, correct? But then if they also have phage receptor protein genes piggybacking, then it's taking away the advantage, right? So not all of them have both. But it's mainly an advantage for the phage to be able to have plasmids that can jump from bacteria to bacteria and confer susceptibility to the phage. Not good for the bacteria, of course. Michelle wants to know if a 2-month-old had a serious reaction to Tdap. He's 23 now, trying to catch up with vaccines. Should he avoid this one if there's an alternative? I think you have to talk to your physician. I'm not a medical doctor, and I don't want to give medical advice. And you have to be careful of saying that Tdap did that also, right? Because you just don't know. Margot, I don't know what being really smart has to do with it at all. It's being able to look at information and make your own conclusions. Strokes, myocarditis, perhaps, not strokes. Do no harm. Primula non nocere. Unfortunately, far too many medics refuse to wear N95 despite vast hospital infections. Artemis wants to know, why does Europe have a smaller recommended number of vaccines for children than the U.S.? Children there don't seem to get sicker. I don't know. There must be an explanation, Artemis. I don't know what it is. It may be combination vaccines, for example. I don't know. How do you think this 1970s flu situation compares to the current moment with H5N1? Some of my friends really want an H5N1 vaccine rollout. No comparison. The 1976 swine flu was not circulating in anything. So there was an outbreak at Fort Dix, and that was it. It's not circulating in people. Maybe it was in pigs somewhere, but had been in pigs forever. And there was no global circulation as there is for H5N1. There were no lethal cases as there is for H5N1. So I think they're totally different. Some of your friends really wanted a vaccine rollout. You can't, because there's not enough human disease to justify a vaccine rollout. And, that's what they did in 1976. They decided to roll it out, and they predicted there would be disease. There wasn't. People got Guillain-Barré. They were really pissed off. You can't do that. You have to wait, unfortunately. Pete wants to know, what's tonight's mini lecture? Okay, so that's a cue. Enough discussion for now. Let's take a break and have a mini lecture. So what is this mini lecture about? Genomes and genetics. Let's move it over here. Maybe make it a little bigger. Nope, too big. Okay. A genome is the nucleic acid of a virus, right? In the 1950s, the breakthrough came that the nucleic acid of the virus is the genetic code. In 1944, it had been discovered that the DNA of bacteria was the genetic code. And then in the 50s, that was extended to viruses, using two viruses, a phage and a tobacco mosaic virus. And many of you certainly learned about the Hershey-Chase experiment at some point, where the question is, in a bacteriophage, which is composed of protein and DNA, what's the genetic material? Many people felt that protein was the genetic material because there are 21 amino acids as opposed to 4 bases that make up nucleic acids. It couldn't possibly be the genetic information. It's not complicated enough. So Hershey and Chase did this experiment where they have 2 preparations of bacteriophages. These are viruses that infect bacteria. The first you grow in the presence of radioactive sulfur. So that means the proteins are radioactive, and you can measure them in a certain way. And then you have phages grown in the presence of radioactive phosphorus, and that labels the DNA. So the sulfur labels the protein, the phosphorus labels the DNA. So you allow the phages to attach to bacteria and inject their nucleic acid, and then you put them in a kitchen blender and you shear off the bacteria after like 5 minutes, and then you let the infection go and you ask, where is the radioactivity? No radioactivity is detected in the next generation of phage, but you do find radioactive DNA. So this proved that the DNA was the genetic material. Very, very famous experiment. A similar one was done with RNA viruses. The 2 individuals here were Frankel and Conrad. They had this. So tobacco mosaic virus is a RNA virus that infects tobacco, right? And they had found 2 strains, and we'll simplify by calling them the green strain and the red strain. And they were able to disassemble the particles and purify the protein and the RNA. So they did a mixing experiment because they want to know, where is the genetic information, in the protein or the RNA? So they mixed red protein with green RNA, and that produces new viruses. They assemble into virus particles, and then they mix green protein with red RNA. They assemble into particles. Then they infect tobacco leaves. Lo and behold, the protein is always the color of the RNA. So the green RNA encodes green protein. The red RNA encodes red protein. So again, in this case, RNA is the genetic material. But the bigger surprise is that even though there are thousands and thousands and thousands and thousands of different kinds of viruses, there's a finite number of viral genomes. There are only 7. And if you live in New York, you can remember that by the number 7 train icon. There it is. If you don't, it means nothing to you. But I teach students in New York, so it means something to them. There are only 7 different kinds of viral genomes. And all of these genomes have to make mRNA that can be read by host ribosomes. Every virus has to do this because viruses don't encode the complete protein synthesis machinery, so they have to depend on the host. So no matter what kind of genome they have to make mRNA that can be translated by the host. So David Baltimore, who won the Nobel Prize in the 70s for discovery of reverse transcriptase, and who was my postdoctoral advisor, he used this insight that mRNA has to be made by all viruses to categorize viruses in a simple way. He made this called the Baltimore scheme, where he put mRNA at the middle, and then he put the 7 genomes around, actually 6, because at the time he didn't have the 7th group. But double-stranded DNA was group 1. Single-stranded DNA was group 2. Double-stranded RNA 3. Plus RNA that goes through minus RNA 4. Minus RNA 5. Plus RNA that goes through DNA by reverse transcription 6. And finally, 7, the gapped DNA of hepatinoviruses that encode a reverse transcriptase. So all of these have to make mRNA. And that's a simple way to categorize them. Now, before we go any further, some definitions. And we've been talking about this for a while, but just to re-emphasize, when I talk about plus and minus strands, a plus strand of mRNA, well, mRNA is always the plus strand. We just give it that name randomly. It doesn't have anything to do with electricity or polarity. Just plus means it is mRNA. The DNA of the same polarity would be a plus strand as well. The complements of plus strands are called negative strands. And that's how we call plus strand RNA viruses and negative strand RNA viruses. But not all plus RNA is mRNA. The retroviral mRNA genome is plus-stranded, but when it gets in the cell, it is not translated into protein, so it's not mRNA. So you have to be very careful. So the elegance of this system is that if I tell you, here we have a virus with double-stranded RNA, you can tell me the basic steps that are needed to produce mRNA. So, for example, if I say, okay, take a single-stranded DNA, what does it have to do to make mRNA? Okay, so first of all, single-stranded DNA can't be made into mRNA. It has to be double-stranded. So the single strand has to be copied, and then the double-stranded DNA can be made into mRNA. Similarly, the gapped DNA of hepatinoviruses, which we should do a mini lecture on at some point, very important viruses, can't be made into mRNA. It's broken. It has to be repaired and made fully double-stranded to make mRNA. So you can see for every one of these 7 classes of viruses, you can track the steps to produce mRNA. So 7 classes of genome: double-stranded DNA, gap double-stranded DNA, single-stranded DNA. Those are your 3 DNA genomes. Then we have double-stranded RNA, plus and minus RNA, and then a plus RNA with a DNA intermediate. Those are the 7 classes. No more, no less. Haven't found any since the 70s when Baltimore did this. Okay, so the viral genome, right? That's important. It encodes the virus. What's the biggest ones? So I thought a table showing you the biggest ones would be cool. So the biggest one we have so far is a Pandora virus, 2.4 million base pairs of double-stranded DNA. It's huge, encoding 2,500 proteins. And then we have another Pandora virus, is 1.9 million. Here's the Mimi viruses, also over 1 million. And just for comparison, a bacterium, Haemophilus, its genome is 1.8 million bases. So these viruses have bigger genomes than this bacterium. All right, so those are the 10 largest virus genomes. Then largest RNA virus genomes. This is going to change. There'll probably be bigger ones. You have 41 kb for these are all nidoviruses, which are related to the coronavirus. As you can see, 41, 35, and 33.5. But look, 41 kb. It's 10 times. Well, not quite, but it's 41 kilobases. 41,000 bases. This is 2.5 million bases. Oh man, it's so RNA is very fragile and prone to mutation. That's why they don't get very big. Although they can get respectable. Okay, smallest known viral genomes. There's some viruses that are called satellites. They depend on other viruses to allow them to replicate and to provide a capsid to encase their genome. Those are called satellites, and they don't encode any protein. They have a genome of 220 bases of RNA. Hepatitis delta RNA is 1.7 kb. Then we have a bunch of small viruses with various lengths of DNA and different numbers of proteins. And here's hepatitis B virus. So these are all, there's a mix of DNA and RNA viruses here, the smallest known viral genomes. They don't encode a lot of proteins. So it's very interesting that viruses that encode a couple of proteins and viruses that encode thousands of protein, they both can infect a cell and wreak havoc. It's very amazing, isn't it? And it makes you wonder why. Well, you can't say why. What's the function of having all those extra proteins in those big viruses? We just don't know. Let's go through these 7 classes of genome and talk a little bit about the viruses. So these are viruses with double-stranded DNA. And we divide them into 2 classes. So here's the double-stranded DNA from which mRNA is made and proteins that can be used to build new virus particles. Some of these viruses, their genome is copied by the host DNA polymerase, like polyomaviruses, SV40, circular double-stranded DNA. But then the viruses with longer genomes, adenovirus, herpes viruses, pox viruses, they encode their own DNA polymerase. They have big genomes, 30 to 50 kbp and 130 to 375 kbp. So these include adenoviruses, herpes viruses, papillomaviruses, right? Human papillomavirus, polyomavirus, and pox viruses. And then here's a giant virus, a pithovirus. Was that on our table? Let's see. Yeah, it didn't even make the top size genome. But this is not even drawn to scale. I wouldn't be able to fit it on the same slide. It's so much bigger than these other DNA viruses. All right, what about the hepatitis B viruses? They have gapped double-stranded DNA genome. So the genome is a circle, and it's incomplete. It's got a gap, and it also has a piece of RNA attached, and it also has a protein attached. And the reason why this is so, we'll talk about when we talk about hepatitis B viruses. It's really a fascinating thing. But these DNAs get into the cell repairs them, takes the protein off, takes the RNA off, closes the gap. Very convenient, right? The cell does that. And then that can be made into mRNA to make proteins. And so these are the hepatitis viruses. There are animal versions of this as well, but the human virus is hepatitis B virus. Single-stranded DNA genomes. One piece of DNA actually can be minus or plus. It doesn't matter, because when this DNA gets into the cell says, we don't want no single-stranded DNA here. Only double-stranded DNA allowed in this nucleus. And they fix it, and they make it double-stranded, and then it can be made into mRNA and then proteins and new virus particles. And these viruses include the circoviruses with circular single-stranded DNAs. Here, TT virus, Torqueteño virus. Everybody has this virus in your blood. Doesn't seem to do anything bad, yet we all have it. And then the parvoviruses, there's one B19 that causes a human rash disease, fifth disease, and then there are parvoviruses of dogs and cats that you should vaccinate your pets against, otherwise they can get very sick. All right, next, RNA genomes. So we're finished with the DNA viruses. Up until now, we have thought that RNA viruses dominate the eukaryotic virus sphere. That means all the viruses of eukaryotes. But that's beginning to change. I think we missed them in the bacteria. And I think bacteria have a lot of RNA viruses that we didn't know about. Now, a key here is that mammalian cells have no RNA polymerase that can copy the genome of an RNA virus. So every RNA virus has to encode an RNA polymerase. And that RNA polymerase will copy the RNA virus genome and also make mRNA from it. So double-stranded RNA genomes exist. Rotavirus is an example of that. These are typically segmented. Here's a reovirus with 3, 6, 7, 8, 9, 10 segments. There can be fewer. Some of these viruses have fewer segments. Some of them have more segments as well. These double-stranded RNAs can't be directly made into mRNA. So there is a strand of mRNA in there, that green strand, right? It can't be translated. This whole double-stranded RNA has to be copied by an RNA polymerase to make mRNA, which will then be made into protein. Plus strand RNA is very familiar to most of you because we have coronaviruses there, and we have flaviviruses like West Nile virus and yellow fever virus, picornaviruses like poliovirus, and togaviruses. And these all have plus-stranded RNA, single-stranded of various lengths. And when these RNAs get in the cell, they can be directly translated into protein to make more virus particles. And of course, to make new genomes, they're copied into a minus strand whose only function is to serve as a template for more plus strands. And here's some examples of plus-sense RNA viruses. So we have our picornaviruses, polio and rhinovirus, the caliciviruses, norovirus, which many of you are experiencing as a plus-stranded, single-stranded RNA virus. The coronaviruses, SARS-1 and 2, MERS, etc., flavies, yellow fever, West Nile, Hep C, Zika virus, they're all plus-stranded RNA. And the togaviruses include rubella virus and equine encephalitis virus. All these have plus-sense RNA genomes. And a very special family because it is a family of viruses with a single-stranded plus-sense RNA, but it goes through a DNA intermediate. And there's one family, retroviruses, that contains these, and they have many human and animal viruses in them, but there are 2 human pathogens, HIV, 1 and 2, and HTLV, 1 through 4. And the strategy is as follows. The genome is plus RNA. It gets into the cell and is immediately copied by reverse transcriptase, which is present in the virus particle, to make single-stranded DNA and then double-stranded DNA. So the plus RNA is never made into protein. That's why I say not all plus RNA is mRNA. And then, of course, the DNA integrates into the host cell, and there it can be made into mRNA to make new virus particles. It's really remarkable. And we've talked about this before. Then we have minus sense RNA viruses. These can be segmented like influenza viruses, where there are 8 segments of negative strand RNA. Or they could be non-segmented like the measles viruses or the rabies viruses. The minus RNA, when it gets into a cell, cannot be translated into protein, right? By definition, it's a minus strand. It can't be translated. So what has to happen? The virus particle has to have an RNA polymerase in it that can copy this RNA to make mRNA that can be used to make proteins and further the replication cycle. So the minus strand RNA viruses have to carry a enzyme in the particle. And some examples of these, the paramyxoviruses, measles and mumps, rabies, a rhabdovirus, the filoviridae, Ebola and Marburg virus, influenza viruses, and arenaviruses, Lassa virus. So these are viruses with minus sense RNA genomes. And I have this little book here. A picture of the cover is Fever by John Fuller, published in the 60s, and told the story of Lassa virus discovery in Africa. And I read this as a young student, and it hooked me to do virology. This is why I am a virologist. I read this book after I graduated college. Really good book, by the way. Now, if you have a segmented RNA genome, right, some of these minus strand viruses have segmented genomes, you can do something special. You can do reassortment, which means your genome is in pieces. When, say, 2 different influenza viruses infect a cell, like H5N1 and H1N1, all of those RNAs are going to be made, and they're all shuffled throughout the cell. And then some viruses are going to have a mixture of them. So here you have R3 is a recombinant or a reassortant virus where all the RNAs are blue. That's because they're from one parent, and one RNA is red. It came from the other parent. So that gives you new viruses with potentially new properties. And when one of the worries about H5N1 viruses is that they will infect a host that's infected with another influenza virus. And this kind of reassortment will occur that could lead to a virus with different properties. And finally, we have viruses with ambisense genomes, which means they have parts of the genome that are plus and parts that are minus. And these include the arenaviruses. These are classified technically as negative strand viruses because they have an RNA polymerase in the virus particle, because this RNA, when it gets into the cell, cannot be translated to do everything. So it has to have an RNA polymerase in the particle to make the proper mRNAs. Okay, I want to end up this mini lecture with a little discussion of how we do genetics of viruses. Now, the plaque assay, a method for quantifying infectivity, was also used to do genetics. So here on this plate, you can see a smaller plaque, and you could pick that and say, does this have some interesting different property? You could mutagenize viruses and plaque them out and look for viruses with different properties. But we don't really do this anymore. We do it by a different way. But before I tell you about that, I want to give you some definitions, okay? First of all, a mutation is a change in DNA or RNA. Nucleotide addition, deletion, rearrangement. It is not a change in protein. A mutation may lead to amino acid changes, but it is not. The amino acid change is not a mutation. A mutation was defined as occurring in nucleic acid, but people use this wrong all the time today. Okay, in the old days, we would mutagenize a viral genome by UV or by chemicals and look for mutants with interesting properties relevant to our studies. But nowadays, we don't do that anymore. We have infectious DNA copies of viral DNAs or RNAs. So we make a DNA copy of the viral genome, and we put it in cells, and out come viruses. It's really a validation of the Hershey-Chase experiment, right? The DNA or the RNA is the genetic material. And you can change this DNA in any way you want. You can add, delete, substitute, make all kinds of changes. You could add genes to the virus. You could remove genes, and you can make them into vectors. And so this is the power of being able to have infectious DNA copies of viral genomes. You can do all kinds of studies. Now, this was first shown for poliovirus, where if you take poliovirus, of course, you infect cells, it will initiate an infection. If you extract the viral RNA and introduce the RNA into cells by a process we call transfection, that will also initiate an infectious cycle because the RNA can be translated into all the proteins that are needed to make new virus particles. If you take the viral RNA and make a DNA copy of it and put it in a bacterial plasmid to propagate it, you can transfect that into cells and viruses will be made. Or you can make RNA from this plasmid in vitro, and those transcripts are also infectious. So this was the first infectious DNA copy of an animal virus, and since then many other viruses have done the same thing. It's the way to study the genome. It's been done for influenza viruses, where there are 8 segments. And so what you have to do, interestingly, is you make 8 plasmids encoding each of the 8 genes of influenza virus, and you've designed the plasmid so that you make both plus and minus RNA from the same plasmid. You put those into cells, all 8 plasmids, and out comes influenza virus. And we use this all the time to modify influenza viruses. It was used to recover the 1918 influenza virus. Remember, that pandemic killed millions, but we didn't have influenza virus, until 1933. But many years later, the viral RNA was isolated from autopsy blocks that the Army had kept from victims of 1918 flu, also from a frozen body that had died of flu. And the genome sequence of all 8 segments was determined, and the virus was recovered by making 8 plasmids and putting them into cells. And this work was led by Jeff Taubenberger, who I interviewed for TWIV, and you can find that episode here. Really interesting to hear about how it was done. Now, this, of course, got people very upset because they said, oh, you're bringing back the 1918 influenza virus. And so it ushered in an era of being suspicious of what scientists can do. You don't always have to clone the viral DNA in a plasmid. Here's an example of the rescue of a virus called horsepox virus. It's a pox virus that is extinct, but we had the genome sequence. So 10 overlapping DNAs were synthesized at a cost of $150,000, put into cells, and out came infectious virus. So you don't have to put the viral DNA in a plasmid. You can just synthesize it. So in this experiment was met with headlines like this one: scientists bring back extinct horsepox virus in lab, raising important biosecurity questions. So, having infectious DNA, and it allows you to do experiments that are not previously possible. So you have to be careful. You have to do them under the right conditions. And in the U.S., we have the National Science Advisory Board for Biosecurity that provides advice regarding these kinds of experiments. And in particular, you may have an idea for an experiment that's perfectly reasonable, but it could have dual use. In other words, someone could use the results for bad purposes. So this committee will help decide if that's the case, and if so, should you do the experiments, and if so, under what conditions. So that brings us to a definition of gain of function research, because one way to do gain of function is via infectious DNA copies. So gain of function, the definition is here. Senators and congressmen giving a new property to an organism. It could be a bacteria, yeast, virus, plant, an animal. Doesn't matter. A new property, not a bad property, folks. It's just a new property. So if you say, oh, they were doing gain of function research in Wuhan, so what? Doesn't mean that it was bad. Now, for viruses, before recombinant DNA, we could give a virus a new property by mutagenizing it, by passing it into different hosts. But today we modify the infectious DNA of the virus. And so what they were doing in Wuhan was taking SARS, they were taking a bat SARS-1-like coronavirus and putting spike protein genes into it from other SARS-1-like coronaviruses that they found in nature. So this was potentially gain of function if you change the property of the virus, but not necessarily. It might not give it a new function. So, this is SARS-1, and the congressional report indicts that as a smoking gun, but in fact, it's not at all. It's if you understand what gain of function research is, it's meaningless. But everyone's confused about it. Here's risky gain of function studies need greater guidance. Really, they're not all risky. And this one, fight over COVID's origin renews debate on risks of lab work. This is, again, involving gain of function. NIH documents provide new evidence U.S.-funded gain of function research. So what? So what if they fund it? It has nothing to do with SARS-CoV-2. Republicans spin NIH letter about coronavirus gain of function research. Basically, when you mix science with politics, you get politics, because politicians either don't understand the science, or if they do, they want to use it. They want to twist it to their advantage. Okay. I'm sure you have plenty of questions. Thank you for listening. Gain of function is just a tinderbox. Okay. Thank you, Raise11, for your contribution to science communication. Interesting. Abhilash says, would we expect better correlates of protection and decreased transmission if seasonal flu vaccines were administered intranasally? Well, some of them are, right? Flu mist is administered intranasally, and overall it is no better than the inactivated vaccines. So I don't, unless you could formulate it differently, I don't think it would be better. We'll remind you to watch my forthcoming virus lectures. So they start next week, especially episode 1, which is pretty layperson friendly. Lori ran into an old friend. Within 5 minutes, it was obvious she was anti-vax. And if you dug deep, she wouldn't really know why she was anti-vax. Tom says, my pal, an MD in Wyoming, it breaks my heart when I listen to lungs of an infant with whooping cough. Yeah. Louis says, what do you think about treatment of flaviviruses with small molecules targeting structural proteins instead of non-structural proteins like proteases? I think it's great. There are a couple of examples of antivirals now for HIV and Hep B that target structural proteins. And they're great proofs that can be a viable target. You don't have to do an enzyme. So, yeah, it's cool. Very cool. Margot writes, I wasn't sure when mRNA vaccine was first scheduled to be released. That's when I found you on a video. Then I felt like I understood. Thank you, Margot. I appreciate that. Do you know universities that do research into baculoviruses? Yeah, for sure. Let's see. I was at one in Kansas, University of Kansas, but I don't remember which one. But there are plenty. You can just go to PubMed and search for papers, and you'll find them. But yeah, she has. There are plenty of baculovirus labs. Yeah. Why are lentivirus infected cells not considered GMO? The DNA is altered, right? Well, they are. I mean, that's the thing, that GMO is not just something scientists do, right? Nature does GMO all the time. So that's one example, but there are many examples of recombination in viruses and bacteria and fungi and parasites where you have different organisms made. Nature is a very good genetic engineer. So yes, GMO is not just done by people. Yep, GB has also been a side effect of Zika virus infection. Yeah, it's good. Lucky that none of the phage capsid proteins are phosphorylated. That's a good point. That's the Hershey-Chase experiment. Or there are some phages where the proteins get in as well as the DNA, right? So if they had picked that phage for their experiment, they would have been in trouble, right? But, luck. Yes. Yes, lovely use of sulfur and phosphorus to discriminate between DNA and proteins. It's the classic experiment. It's great. Pete says the plus DNA produced the plus RNA copy or its complement. The minus DNA produces the plus mRNA. Yeah, the plus DNA doesn't make any RNA for the most part. Yeah. Sharon is in Tasmania. Thank you for your 2025 wishes. Welcome. Try and ask if the porcine encephalomyelitis and hemaggglutinating virus, what is going to be the mechanism of hemagglutination? Probably it encodes a protein that binds sialic acid, a glycoprotein of the virus that binds sialic acid, and that aggregates red blood cells. Gap DNA viruses are like a wounded Trojan horse. Does that analogy work? Yeah, I mean, it's wounded and it comes in the cell, and the cell takes pity on it and wants to fix it, and then it's a problem. Yeah, I like that. Yeah. Canine parvo is a horrible thing to see. I've never seen it. Take your word for it. And a shout out to norovirus, which many people have had. Yep. Yep. SARS-CoV-2 does not have a HE, a hemagglutinin esterase. No, it does not. Yeah, and gain of function doesn't always mean gain of fitness. It's usually the opposite. Often, yes, when a new property is acquired, it can cause loss of fitness, especially when humans do it, right? Tom writes, golly, Vincent, who developed recovery of viruses in cell culture via transfection of DNA plasmids? Anybody know? Let's see if anyone knows. I know you know, Tom. The Hurst reaction, that'd be hemagglutination, right? We used to do it at room temperature. It would change. Its kinetics would change, and if you do it at too high a temperature, the neuraminidase would reverse the hemagglutination. So it's best to do it at room temperature. Yeah. Microbe, do you think mad scientist movies hurt the public perception? I don't know. It's a good question. I mean, mad scientists have been around. Mad scientist movies have been around for ages. I suspect that at some point people realized they were science fiction, but Contagion, I think a lot of people thought it could happen, and then in a way it did, right? Yeah, the Philadelphia had a yellow fever outbreak. Yep. hmm Steve wants to know about inactivating using far UVC light to inactivate viruses. Oh, it works very well. In fact, we had a guy from Colombia on Twiv a long time ago, or early in the pandemic, to talk about this. And they've been deployed in places where you can turn them on at night, right, when there's nobody in the building, and it will inactivate viruses. And then you come in the morning, turn them off. They're not a scam at all. They work. The public should demand a third house of Congress for scientists. Never going to happen. People don't like scientists enough. They don't trust us. Will, am I correct in assuming that natural mutation of viruses, which happens all the time, inevitably involves gains of? Yes, absolutely. That's how you get new viruses with new properties, like the ability to infect humans. Oh, let's go through this. So there's a virus circulating in bats, 2018, 2019, and it's reproducing in the bats. It's a coronavirus, and it's mutating, and then these mutations randomly make it able to infect people. But the virus, the bats never see people, so it doesn't do anything until the bat infects another animal, and that animal is taken into the market for sale. And so the ability to infect human was a gain of function mutation. Nature is the greatest gain of function engineer, no question about it. Yes, Gwen. So we had a policewoman write in asking about protection against COVID. She said, "My only protection are gloves and a Glock." And I'll never forget that. It was so great. And then in last year, 2024, she said, "Hi, it's me with the gloves and the Glock. I'm still here." Because we always used to speculate, what happened to her? Is she okay? And she had some other question. It was just great. I loved it. John says, "Is the answer our host?" It is. I was the one who developed the first DNA copy of poliovirus, which you could put in cells and recover viruses with. Yeah. Is multiple infection with herpes virus is possible? Yeah. You got herpes simplex, you got Epstein-Barr, you have cytomegalovirus. Yeah, you got them all. You got about a dozen herpes viruses. Latex gloves and a Glock. Yep. Wow, it's amazing you remember that. Yeah, the dastardly furin cleavage site, which was acquired somewhere in nature, right? We now know that there are coronaviruses with such sites in them. So that was a random event, and that's important for infecting people. On the subject of bioweapon labs, I think I remember Alan Dove saying something like, the superpowers found out it's a lot cheaper to use bombs and ammo, and those don't turn around and come back. Yeah, bombs are the best bioterror weapon. Viruses are not, because you really cannot control them. So you can't forget about bioweapons. The Soviet Union tried a lot, and they all fail. And you can read a book by Ken Alibek, Bioweapon. So he was a former worker in the Soviet Union. And he says, for a virus to be a weapon, first of all, they're stored lyophilized. They're stored dried. And when the Kremlin says, throw this bomb on the U.S. with Ebola virus, then you have 60 minutes to reconstitute the dry powder into an infectious form and get it into the warhead. Can't be done. Can't be done. Failed. So that's why there are no viral bioweapons. Now, you could do it differently. You could You can infect someone with smallpox and let them walk into New York City and hang around for a few days. They could, you can imagine people would let that happen to them. But that's just that's just smallpox virus. But you can't get it anymore, right? Because it's restricted. And that's one of the better ones to use for that. Ebola, not so good because it doesn't start transmitting until you get sick. And so you're going to get sick, you're going to be hospitalized, and that's going to limit your transmissibility. So all these reasons why they're not good bioweapons. You can speculate all you want. You can come up with scenarios. But sorry, folks, bombs are still the best. Still the best. Okay, folks. Are you exhausted of your questions for tonight? I'll give you a couple of minutes to. These last two I don't quite understand, so I'm not going to read them. Oh yes, Griffin says spore-forming bacterial bioweapons, however, still not as good as bombs. Yeah, you could do anthrax, I guess. It takes time, right? It takes time. Not good. Not good. Bombs are really the best. Soviet Union learned that. And I spent a lot of time thinking about it and can't really come up with anything that would be effective. What's the latest news on H5N1 flu? A lot of cows in the U.S. are infected. They aren't anywhere else. There's a lot of virus circulating. And in Canada, there's no cow flu H5N1, but there is the bird flu H5N1. So we are thinking, we're just waiting and watching. No, I don't mind. I don't mind going another 15 minutes. It's good. I like doing this. What's my favorite food? I think pasta is my favorite food. Yeah. When's the virology course starting up? Next Wednesday. So Monday is Martin Luther King, and then Wednesday we start up. Shoes off in the house. Yeah, I generally I take my shoes off in the house, mostly to feel like I'm kicking back, right? In the summer, if you have light loafers on, I think that's fine. Although I think you shouldn't go in the bedroom like with outdoor shoes. But in the winter, I wear boots, and I come in and sit in a chair and pull them off because I can't get them off. Otherwise, they're too hard to get off. But they can be muddy and snowy, so you want to take them off. Yeah. Do you think they'll find a remedy for long COVID brain fog? Hard to predict, right? But a lot of people are working on it, and it may take time. But if you can pinpoint the cause, you might be able to if there isn't permanent damage done, right? Yeah, our military found how easy it was to flood the subway with B. subtilis spores and douse San Francisco with Serratia marcescens. Yeah, they did that because Serratia is red and you can easily pick it up. Anyone know a clearinghouse to write to senators about RFK Jr.? Les, can you grab the link that we had put up for safecommunitiescoalition.com and put it up here for Scott? Because, yeah, it's a good site. Hello, Ripticomp from Taos. You're going to have to watch it because we're almost done here. The kid wants to know what type of pasta. I really prefer spaghetti, not angel hair, not linguine, spaghetti with red sauce. Yeah, that's my favorite. And yeah, I love it. Years ago, I went to Italy and some days I ate pasta twice a day. John was looking at early microscopes from Leeuwenhoek. Have you ever seen one of those like in a museum or science? Friend with it as a collectible. I haven't. Of course, I've seen pictures of them. I've never been to a museum that has them. But of course, in the Netherlands, there must be, right? Because that's where he was from. Lollipop is noticing a great deal of COVID. Yeah, so this is the season. The cases are going up because people are traveling, and it's cold, and it's the respiratory season. But they're largely mild, although people are still dying of them, in particular people who are older with comorbidities. But it's like influenza, right? We vaccinate against influenza, we vaccinate against COVID. You're still going to have infections because the vaccines don't prevent infections. They prevent severe disease. So you're going to get infections and mild disease, and that's what we're seeing now. Will your Columbia Virology students be tuning into after hours, office hours? Some of them do. I do at office hours for them in the morning, right, where it's just them. But I'll certainly tell them about it. And in the past, some of them have come. Yeah. I mean, it would benefit them, right, to do that. Leo, thank you for your support of science communication. Really appreciate it. Did you ever get that microscope up to Coxsackie? No, it's sitting here on the floor. You know, I have to build a crate that's going to hold it. It's really heavy and needs to be a wood crate. And I just haven't had a chance. And then Elena, I believe her name was, volunteered to come get it, but it's still sitting here. But I don't think she's listening anymore. Did you see the thing about Alzheimer's and plaque? Okay, so there are two papers that are very interesting. One says that head trauma, which is a risk factor for Alzheimer, activates viruses, and that could be involved. And there's another paper saying that, there's a lot of evidence that herpes virus reactivations in the brain are risk factors for Alzheimer's. And there's a paper, which we may do on Twin, which talks about that. It's very cool. Counting down 15 minutes on a COVID test. All right, good luck. We hope it's negative. How did Typhoid Mary make peach ice cream in New York in the 1900s? No idea. No idea. Fusilli is good too. Yeah, it's a good one. But I like spaghetti. Pasta carbonara is really good too. Yeah, but it's kind of rich, right? I like, there's another one where, yeah, okay, so it's called cacio e pepe, cheese and pepper, spaghetti with cheese and pepper. Cacio e pepe. That's also very good. Why isn't oseltamivir an over-the-counter drug? Yeah, it really should be, right? Because by the time you get a prescription, it's too late. Yeah. I don't know why. Maybe they're worried about it being abused, but in many other countries, it's OTC. Yeah. With respect to shoes, Wendy writes in the house, my dad, an ACOM histology professor, Albert Einstein College of Medicine, used to proclaim, no bare feet. The amino acids from sweat eat up the carpets. That's very interesting. I didn't know that, Wendy. Very cool. Always grateful for the education that you give us, but I get education from you as well, like no bare feet on the carpet. Vincent calls it sauce and not gravy. I thought most Jersey Italians called it gravy. Yeah, I always called it sauce. I don't remember what my grandmother used to do. So my grandma and grandfather and my father were all immigrants. I think she called it gravy, but it didn't stick with me because for me, gravy is something else, right? Red sauce, yeah. Cool. Pete said, my first month at uni in 1972, my physics tutor sent me off as an exercise to research Leeuwenhoek. The 1670 copies of the Royal Society were on open shelves at Imperial to read his paper. That's so cool. That's very cool. Yeah. Gwen is going to come get the microscope. Yeah, if you want to come and get it, I mean, you're far away. But if you ever come to New York, you're welcome to it. Lise, I love cacio e pepe. Yeah, but it has to be done properly. It's not easy to make. Yeah. Heather finished spaghetti for dinner. Wonderful. Love it. Lollipop asks, so are healthy younger people under 50 who are not vaccinated, who get COVID, still at same risk for develop serious disease? Haven't they had exposure? They're not as at risk as they were right early in the pandemic. However, they are at risk for long COVID, and vaccination does reduce frequency of long COVID. So that's, for that, it's worth getting vaccinated. Scott says, I've been wondering if long COVID research has seen any impact on mitochondrial function. My SIL is now nearly 5 years with debilitating LC. I'm sorry to hear that, Scott. Yes, there's a lot of interest in mitochondrial function for sure. Yeah. Right, here's the spelling, cacio e pepe, right? Okay, here's the place to go write a letter to your senator is in this video here. Questella, thank you for your support of science communication. And Child, hello. Thanks for coming by and watching. Love that spaghetti. Will says, I've always enjoyed pasta, but living in China, I prefer their noodles. Was it Marco Polo who brought back pasta? Yeah, I think so. I thought he brought it back from China. Yeah, I can see where you might like it, but I prefer, I love cold noodles with sesame. This is my absolute favorite Chinese dish. It's a different noodle, but it's not like spaghetti. The pasta. It's different composition, obviously. All right. Yeah, Beyond the Noise is up. Scott, thank you for your support of science communication. Really appreciate it. I'm going to find the link right now. What is it called? Safe Communic. Yeah, here we go. I'm going to paste it into the chat. Here it is. That's the place to go to send a letter to your senator to not vote for RFK Jr. as head of HHS. All right, folks. Thanks for coming tonight. Really appreciate it. Nice conversation. Good questions. ChatGPT says pasta in Italy predates Marco Polo. I don't know. Should we trust ChatGPT? Tom and Les and Barb Mack, UK, thank you so much for moderating tonight. Really appreciate your work. Thanks to all of you for coming and participating and asking good questions. I think you're all fantastic. We will be back next week. I'm trying to do this every week now because there's always good things to talk about. And as we get towards the hearings and so forth, we'll be talking about that as well. So the next one is Wednesday, the 22nd of January at 8 p.m. Eastern. Yeah, sauce sticks really well to fusilli. I know. Yeah, we could have a whole discussion about pasta. There are a lot of other quirks and quarks, but for now, thanks everybody for coming, and see you next week. Bring back your always good questions, and then until then, of course, I want you to stay safe. Be careful. Good night.